Alzheimer's disease vaccine is really thinking ahead to the future. There are therapies being developed for Alzheimer's disease, promising antibody therapies, but thinking ahead, it would be very convenient to have a vaccine where the patients would make their own antibodies. So the project that we've undertaken is to make a vaccine that would contain epitopes, so that's a portion of the amyloid-β protein that drives disease in Alzheimer's disease, and put that into a vaccine. The advantage that we have is that we have a platform to determine when amyloid-β misfolds and forms aggregates, small oligomers, little clumps that are toxic. There are new shapes that appear, conformational epitopes, and we can determine where these are. We can reproduce the shape and put that into a vaccine so that when you immunize, you're generating antibodies only against the toxic form of A-beta without interfering with normal A-beta that the cells need. In terms of development, this is an early stage project. So right now we've done experiments in mice, and we've been able to see that we do indeed elicit a good antibody response in mice using a carrier protein, so something that we attach our little epitope to, and adjuvants to enhance the immune response that are compatible with human use, so that could be used in humans. And we also were able to determine that when we immunize, we don't elicit a detrimental immune response, that is a T-cell immune response, which in prior vaccines has been an issue. When T-cells recognize an epitope in A-beta and then see it again in the brain, there was inflammation, and the trials had to be stopped because of safety reasons. So the way we design our vaccine, we avoid stimulating the T-cells, only the B cells that make antibodies. So the next steps are to optimize the formatting of the vaccine and the type of adjuvant, the concentrations, how many doses, optimize the dosing regimen, and then test in a disease model. So not just look at immune response, but look at the impact of the immune response on disease progression in an animal model. And then from that proof of concept, we would move on to the more formal studies that are needed to do a clinical trial in humans.